Today, we're diving into the microscopic world to tell a story. A tale of two bacterial cousins. They are so closely related, they can fool even the most experienced scientists. But here's the thing. One is a killer, and the other, well, it pretty much just hangs out. So let's get right to it. You've got two bacteria from the exact same family. One can cause devastating diseases like pneumonia and meningitis. The other is a totally normal, mostly harmless resident of your mouth. With the stakes this high, how on earth do labs figure out which one they're looking at? That is the mystery we are going to solve today. So, to really start our investigation, we've got to understand why these two are so darn hard to tell apart in the first place. And it all comes down to a really strong family resemblance. Okay, so check this out. Both Streptococcus pneumoniae and Viridans streptococci are from the same family. They're both gram-positive cocci, which basically means they're little purple spheres under a microscope after a standard lab stain. They're also both catalase negative. And if that's not enough, when you grow them on a special dish called a blood agar plate, they both create this greenish tint called alpha hemolysis. So you can see how in the first round of tests, they look completely identical. But, any good detective knows you have to look closer. So while they might seem like twins from a distance, putting them under the microscope starts to reveal some tiny but absolutely critical differences. All right, let's break down the visual tells. First up, their shape, or morphology. S. pneumococci is what we call lanceolate. It's kind of pointy, almost flame-shaped. Viridans, on the other hand, is just your classic round or oval bug. Next, how they hang out. Pneumococci are usually in pairs. We call them diplococci, while Viridans likes to form these long, elegant chains, almost like beads on a string. And then there's the capsule. Think of this as bacterial armor. S. pneumococci has one, Viridans doesn't. Even the way they grow on a plate is different. Pneumococci colonies look kind of sunken in the middle, like a checkered piece. And let me tell you, that capsule is a huge deal. It's not just a fashion statement. This slimy, protective shield is exactly what helps S. pneumonia hide from our immune system, making it incredibly difficult for our body to fight it off. Seeing that capsule is a massive red flag that you are dealing with a serious pathogen. Now, visual clues are a great start, but when a patient's life is on the line, you need 100% certainty. And that's where some very clever biochemical tests come in to finally blow their cover and reveal their true identities. First on the list is the bile solubility test. This is just brilliant, really. It turns out S. pneumoniae has a built-in self-destruct button, an enzyme called amidase, and bile salts are the chemical trigger that pushes that button. Viridans streptococci, they don't have this enzyme at all. So the result is like night and day. You take a cloudy tube of S. pneumoniae, you add the bile salts, and the bacteria literally burst open. The cloudy solution turns completely clear. That's a positive confirmation. You do the same thing to Viridans, and nothing. The tube stays cloudy because the bacteria are totally unaffected. Okay, the next definitive test is called Optochin sensitivity. Just picture this. You grow a lawn of bacteria on a plate, and then you place a tiny paper disc soaked in a chemical called Optochin right in the middle. For one of these cousins, this chemical is basically kryptonite. And here is the magic number you're looking for. 14 millimeters. If you're dealing with S. pneumococci, it is so sensitive to Optochin that it refuses to grow anywhere near that disc. It creates this clear, bacteria-free halo around it, a zone of inhibition that has to be at least 14 millimeters wide to be a positive ID. I mean, the result is just unmistakable when you look at the plate. S. pneumoniae is sensitive, so you get that big, clear zone around the disc. But Viridans, it's resistant. It could not care less about that chemical. It'll grow right up to the edge of the disc. At this point, the case is closed. You know exactly who you're dealing with. So, we figured out how to tell them apart, both by sight and by science. But now we come to the big one, the most important question of all. Why does any of this matter so much? This is where the lab work meets real-world health. Meet the foe. Streptococcus pneumoniae is a major league human pathogen. This is the bug behind a really serious lung infection called lobar pneumonia. It's the one that can get into your nervous system and cause deadly meningitis. And it's the culprit behind millions of middle ear infections in kids. Getting the idea on this one, right and fast, is absolutely critical for treatment to begin. And then we have its much tamer cousin. Viridans Streptococcus are what we call commensals. Basically, they're part of the normal team of microbes that live in and on us, mostly in our mouths and throats. Sure, they're a main cause of cavities and in rare cases can cause a heart infection, but their potential for harm is just on a completely different level than the immediate danger posed by S. pneumonia. You know, this simple quote really just says it all. One is a major human pathogen. The other is primarily a commensal. It perfectly captures the difference. One is an invader that our body needs to fight with everything it has, and the other is just a neighbor, usually living peacefully alongside us. So, there you have it. Through careful observation and a couple of definitive tests, scientists can tell a dangerous foe from a harmless friend. The tale of these two cousins is such a great reminder that in the microscopic world, looks can be incredibly deceiving, and getting the right name can literally be the difference between life and death. It really makes you wonder, what other microbial lookalikes are out there hiding in plain sight?